Thank you, Becca. My name is Elise Fauché, and I am from New Iberia, Louisiana. I am an Usher parent to Hunter, who is 11 and has type 1C. My husband Blair and I also have an 8-year-old, Harrison, and a 6-month-old, hot-off-the-press baby girl, Mary Blair. I went to my first Usher conference when Hunter was diagnosed back in 2012, and I've had the privilege to serve on the Coalition's board of directors for a few years as well. I hope you all benefit from this weekend as much as our family has over the past 10 years. I've been knowing Dr. Lentz for probably around 10 years now, and she and Susie Trotochaud are a force to be reckoned with. They are two of the most dedicated people I know working towards a cure for Usher syndrome. Jennifer Lentz is an associate professor of otolaryngology, if I pronounced that correctly, and biocommunications, and a member of the Neuroscience Center of Excellence at LSU Health in New Orleans, Louisiana. Dr. Lentz is also adjunct in the Department of Ophthalmology and Genetics. The overall goals of her research are to understand the mechanisms of Usher syndrome and to develop therapeutic approaches to prevent or cure the deafness, imbalance, and blindness associated with this disease. Currently, her lab targets mutations in the USH1C gene with antisense gene and CRISPR-based therapies using cell lines and a knock-in mouse model of Usher syndrome. Dr. Lentz also conducts several natural history studies, both retrospective and prospective, with Usher syndrome patients to better understand the molecular epidemiology and natural, clinical, and socio-demographic histories of Usher syndrome. Susie Trotochaud is the mother of 2 children with Usher syndrome and the executive director of Usher 2020 Foundation, a nonprofit funding research to find treatments to slow, stop, or reverse the degeneration of sight caused by Usher syndrome and retinitis pigmentosa. Let's welcome Jennifer and Susie.
Good morning. I'm so happy to be here to meet new individuals and to reconnect with old friends. I want to first say thank you for taking the time and making the arrangements to be here with us today. And also, I just want to take a minute to thank all of the interpreters in the room that are making it possible for us to connect. We scientists can get very excited when someone asks us to talk about our work, so we're going to work hard to slow down so that we can connect and tell you about our research. Today I'm going to talk about new therapy development in Usher syndrome type 1C models. First, I'm going to check that I have the pointer and the slide. Yep, okay. So in one slide, I'm going to describe the basics of Usher syndrome. There is a lot of information and even new information. So if you don't hear about a topic you're interested in, or have questions, please reach out to us. We're very excited to meet you and connect with you and hear about your experiences as well. So Usher syndrome is an autosomal recessive genetic disorder. That means you have to have 2 copies of a mutation, one that you get from your mom and one that you get from your dad in order to have Usher syndrome. And the autosomal part means that both males and females get Usher syndrome. The symptoms include hearing loss and vision loss together, and some individuals also have a loss of balance. We use the numbers Usher type 1, Usher type 2, Usher type 3, etc., to describe the severity of the symptoms and the age at which they might onset. So they are a clinical diagnosis. We use the letters then to denote which gene you have a mutation in that is the cause of your Usher syndrome. So for example, individuals with Usher syndrome type 1B have a type 1 clinical course. So they were likely born deaf, have balance difficulties, and begin to lose vision or experience symptoms of vision loss around the age of 10. And the mutations that cause their Usher syndrome are in a gene called myosin 7A. This is USH1B. So the numbers are the type, the clinical type, and the letters are the gene. There's been a lot of progress recently, and a new clinical category for Usher syndrome, a type 4, and also the description of atypical and ultra-rare Usher syndrome. Still, there are individuals that have hearing loss and vision loss symptoms like Usher syndrome, but we do not yet know the genetic cause of their disease. So there will be new types and new genes in the future. So before I talk about our research specifically, I wanted to take a moment to talk about the typical path to progress in understanding the symptoms in Usher syndrome, the causes, and the development of new therapies. Typically, this begins with observations in patients. So it's very important for those of you individuals living with Usher syndrome to see your doctors. We learn about the symptoms that you have, when they begin, and how quickly they progress. We then use this information to develop models of Usher syndrome, cellular models or animal models. And we develop these animal models so that we can do experiments on eyes and ears in Usher syndrome. We also use these models to develop and test new therapies. And then once we have identified a therapy that works well in animals and is safe, we conduct research in individuals with Usher syndrome to test the new therapies. Sometimes these therapies work pretty well, like we had intended them to. Sometimes they work well for some patients, but not for all patients. So then we go back to the drawing board and we study those symptoms again in patients. We make sure we have the right models, maybe develop new models. Maybe we tweak the drugs again and test them again in patients. The goal for those of us in basic science and clinical science doing research is to develop a therapy for each individual with Usher syndrome that works well for them. So now I'm going to talk a little bit more specifically about what we do in my lab. On this slide is a picture of the USH1C gene. It's a sort of roadmap. And what's highlighted above and below this picture of the gene is a list of all of the mutations that we know of, about 50, that cause Usher syndrome. If you have any two of these, it can be the same mutation in both your copies from your mom or your dad, or it can be two different ones, one you got from your mom and one that you got from your dad, that can cause your Usher syndrome. In my lab, we focus on the 216A mutation, which is the Acadian mutation, and Susie, who's going to talk after me, focuses on the 91C to T mutation. We've developed a mouse model with the 216A mutation, and Susie's foundation has funded a research group to develop a pig model with the 91C to T mutation. So the 216A mutation is a splicing mutation. That means it causes a change in the natural way that the gene splices. This is a process that happens where splicing proteins cut out portions of the gene transcript and then stitch it back together to make the final code for a protein. And the 216A mutation affects that process. The picture on your left shows correct splicing. The splicing machinery here is depicted as scissors. So it finds the correct place and cuts out an intron and stitches it back together. On your right, we can see the 216A mutation mislocalizes those splicing proteins, and the cut happens in the wrong place. And that leads to an incorrect protein. In my lab, we developed antisense oligonucleotides to target that 216A mutation and prevent wrong cutting or aberrant cutting and force correct cutting. So we developed a mouse model with the Acadian mutation. We knocked it into the mouse genome, and the mice have deafness. They have much elevated hearing thresholds. The mice also have circling behavior, which is indicative of a balance disorder, and they have mildly reduced visual function. And we use this mouse model to test new therapies. When we treat the mice with the antisense oligonucleotides one time, we can restore their hearing and prevent their circling behavior. You can see in the video of the mice playing, the mouse running in circles is an Usher mouse treated with a placebo drug. So an antisense oligonucleotide that's not targeted to the mutation, and he continues to circle. And the mouse immediately to its right was treated with our antisense oligonucleotide, and he no longer circles. The graph on your right also shows that when we treat our mice one time with an antisense oligonucleotide, that we improve their visual function for at least a year. Now I'm going to turn over the talk to Susie.
Hello. My name is Susie Trotochaud, and I'm the Executive Director of Usher 2020 Foundation. I'm not a doctor or a scientist, and unlike some of the speakers you'll hear this morning, I have no letters before or after my name. I'm just a mom of 2 young adults with Usher syndrome, specifically USH1C. My husband and I started our foundation in 2012 with one goal, as was told earlier, to slow, stop, or reverse the degeneration of sight caused by Usher syndrome. Our funding is focused on research and has gone to support a number of programs, including the NAC Attack Extension Study that led to the clinical trial that Dr. Peter Campanchero from Wilmer Eye Institute has presented virtually at this conference, and I highly recommend you take time to watch that, as that is a mutation-agnostic treatment. And also the ASO treatment and the natural history studies out of Dr. Lentz's lab. We also promoted other programs as well. But for today, I will focus on gene therapy in our large animal model. First, I would like to give you a quick and simplistic overview of what gene augmentation therapy is. This is not to be confused with gene editing or other ways gene therapy is used to supplement treatments. And apologies to all the scientists in the room, but as a layperson, this is how I see it. In the most simplistic way, gene therapy is like a store that delivers its products, say groceries. Is like a store that delivers products, say groceries, to its end point. In our analogy, the store is the lab where a mutated gene, as everyone with Usher syndrome has, is corrected. That gene is then packaged into an emptied out virus or vector, much as groceries are loaded into an empty truck. Just to be clear here, the virus has been rendered harmless of its original effects. It is now just a delivery mechanism. The vector is then placed into the retina of the eye, just as the delivery truck might bring groceries into multiple homes. Once in the eye, the virus does what viruses do. It invades the cell and dumps its cargo. Now the cells have corrected genetic code and should be able to make the necessary protein needed to mitigate the effects of vision loss caused by a specific Usher mutation. Over the past 6 years, through a consortium of researchers from around the world, we have created all the components for a gene therapy program for the USH1C mutation, including the truck, payload, and map. The truck is the capsid. This came out of the work at Luc Vandenberg's lab at Mass Eye near Harvard. We are using a novel capsid called ANC80. However, I would add that our research group is also testing a more commonly used vector, AAV9. The payload includes the corrected USH1C DNA with what our scientists have concluded is the best isoform. And I'm going to leave isoforms for another presentation. Finally, our consortium agreed to test 2 vector plasmids, specifically promoters, head-to-head to determine which one would work best. Again, I'll leave the promoters to another presentation, but for simplicity, imagine that the vector plasmid is like the map used by the truck driver to deliver his load. We are looking for the best map. The only thing left to our simplified gene therapy analogy is the house or delivery point. For this, we created a large animal model, specifically a pig. Pigs are good models for some representation of human anatomy, including the eye. Although not as inexpensive as zebrafish and mice, they are not as expensive as non-human primates, so they have certain advantages. Our pig model has robust hearing, vision, and balance characteristics, as can be seen in the paper published by our consortium in March 2022. The pig is also a good model for testing gene therapy, as it has an eye size that is similar to that of humans. This is important for getting dosing right before starting human trials. Now, getting to the point of testing gene therapy on our animal model has been a long, arduous process. We started with this program with the initial conception in 2013 and development of the animal model in 2017. And since then, our dedicated veterinary group has had to learn to work with, care for, sustain, and breed animals that have the challenges of Usher syndrome. But we now have an established breeding herd at 2 locations. We have completed safety studies of our gene therapy on non-diseased animals, and just yesterday completed our first gene therapy surgeries on animals with USH1C. So this was a big milestone for us. I'd like to introduce you to one of our precious animals. I'm hoping this video works. This is a demonstration of some of the behavior testing done on our pig model. The first video will be a wild type or unaffected pig going through an obstacle course, and the second will be our USH1C affected animal going through the same course, showing the visual loss that is already prevalent after 7 months of age. This is the affected animal. We have a very aggressive timeline to try to get our gene therapy into the clinic, which hinges on concluding our efficacy studies by the fall of 2023. In the next year, we plan to begin talks with the FDA so as to make sure we are following all the regulatory requirements, begin creating clinical grade vectors that can be used in humans as soon as we have identified our lead candidate, and start safety and tox studies again as soon as we have identified our lead candidate. However, we are well aware of the valley of death that occurs when preclinical studies move into the clinic. Believe it or not, we are actually almost through the easy part. But we are doing everything we can to overcome these obstacles and continue to hope for the best. Dr. Lentz and I would like to thank all of the people and organizations that have made this possible, and thanks to all of you for listening. I would like to end with the wise words of Helen Keller. Optimism is the faith that leads to achievement. Nothing can be done without hope. Here's to hoping. Thank you.